It's super nice to see all these familiar faces and old friends. It's nice to see Nancy Moran, Nassos, Enrich, Thomas, a bunch of you, Philippe. So I hope the next years we'll have the chance to interact with each other and start new collaborations. I'm looking forward for this SymbNET network. So now regarding my laboratory, I work at the Gulbenkian Institute, as Miguel said, and in my laboratory, we are particularly interested in understanding the role of interspecies interactions mediated by processes, bacterial signaling processes, such as quorum sensing, and how this molecular process play a role in shaping bacterial communities that are composed of many different species. So in particular, we are interested in what I think is the most interesting polyspecies community that we know of, which is the mammalian gastrointestinal tract, where we have hundreds of different bacterial species interacting with each other and interacting with the host. And very importantly, we know that this polyspecies microbial community that resides in the mammalian gut, it's very important for many host functions, such as nutrition, education of the immune system, or the protective barrier against invasion of pathogens that is called colonization resistance. In my laboratory, to study the functional aspects of the gut microbiota, we've been recently focusing more in understanding the processes involved in colonization resistance, which are the natural ability that the healthy microbiota has to act as a protective barrier against the invasion of pathogens or against the expansion of pathobionts. And the reason why we focus in this particular property of this particular beneficial property of the microbiota, it is because it's here where I think microbe interactions play a more direct role. So to study the role of interspecies interactions, microbe interactions in the mammalian gut and their function in colonization resistance, we've been focusing a lot we've been using a lot a system where we perturb the microbiota with antibiotics. So antibiotics can cause imbalances of the gut microbiota that leads to negative functions related to the host. And so we call this dysbiosis. So we've been using this model to study how antibiotics disrupt the microbiota community to understand the most relevant microbial interactions taking place in this community, their molecular mechanisms, and the functional implications of these interactions. So using this model, we've been focused in the last 5 years or so. We've been focusing mainly in these three processes that I'm listing here. So we studied the role of bacterial quorum sensing, which is a bacterial signaling mechanism that many bacterial communities use. We've established the first model to manipulate quorum sensing in the mammalian gut. And with this model, with this mouse model, we were able to show that we can manipulate bacterial quorum sensing during antibiotic-induced dysbiosis, and this can have consequences for the balance of the most important bacterial phyla in the mammalian gut, which are the Bacteroidetes and the Firmicutes. And during antibiotic-induced dysbiosis, by manipulating quorum sensing, we can counter pathobiont expansion. So it has a functional consequence. Using the same model, we looked for species that have a protective properties, and we were able to identify a keystone bacterial species that has consequences at the level of colonization resistance against Enterobacteriaceae through a mechanism of nutrition competition. And I'll be talking a bit more about this process here. And in collaboration with Isabel Gordo at the Gulbenkian Institute and Jocelyn Demongeau, we've used experimental evolution to study the evolution of E. coli in the mammalian gut. And in particular, I focus in understanding the microbe interactions involved in this process. And we've shown that the gut metabolome, the microbial metabolites produced by the bacteria in this environment are among the most important drivers shaping the evolution of E. coli, and I think other bacteria in the eco-evo interactions occurring in the mammalian gut. So through these studies that I was just summarizing that we've published in the last 5 years or so, I think we can go from this like less resolution type of cartoon to a different type of resolution of cartoon, that now we can put some real chemical molecules in this cartoon that we show to be important either in cross-sensing manipulation with consequence for pathobiont expansion, in the evolution of E. coli through shaping the gut metabolome that has consequences for the eco-evo interactions, and also in colonization resistance by identifying a keystone microbiota species through a mechanism of nutrition competition. So this is kind of like a snapshot of our last findings in the last 5 years. This is all published work, and I welcome you to read more about it if you're interested in learning more. So for now, for the rest of my talk, I'll just focus on this story here quickly. Okay, so as I told you before, we use antibiotic-induced dysbiosis and using a mouse model to disrupt the microbiota community, and then we look for the functional consequences of this disruption and how we can recover from it. So what happens when we treat mice with streptomycin? There's an immediate, very strong consequence at the composition level of the microbiota. This is also a very extended treatment, but what we observed is initially we have the usual nice balance between firmicutes and bacteroidetes in the mammalian gut. Upon antibiotics, which is in this case streptomycin, we see the domination of bacteroidetes, and the functional consequences of this domination are, for example, the expansion of a pathobiont that in our facility it's typically Bacteroides vulgatus, which has been shown to cause inflammation and other problems for the host. So there's a very strong pathobiont expansion, and there's a very strong loss of colonization resistance against pathogen. Here we are just looking at E. coli colonization in the mammalian gut. If we have untreated animals, the healthy microbiota provides colonization resistance. So even if you give a very high load of E. coli cannot colonize these mice. But if we treat the animal for 2 days with streptomycin, and then you continue to the antibiotic treatment, an E. coli that is resistant to streptomycin can colonize to very high levels and persist in the mammalian gut. So antibiotics increase host susceptibility to infections and break down colonization resistance. So my student Ana Rita, she was looking at what happens after stopping the antibiotics. So she did an antibiotic treatment for 2 weeks, and then for 6 weeks she stopped the antibiotics, and then gave E. coli to these mice that had been subjected to the antibiotic and to recovery from it. And then what she did was to score the E. coli loads that would be, were able or not to colonize these mice. And what she observed was the huge heterogeneity in the colonization levels of the E. coli of these mice after antibiotic recovery. So if you look at the dark blue lines, what you see that in some mice there's really no colonization resistance. But for example, in these mice here, there were several, like 3 orders of magnitude lower colonization of E. coli. So this mouse here has much stronger colonization resistance capacities than these mice here. So given this heterogeneity, it was not very difficult for Rita. So what Rita decided to ask was, what are the microbes that are responsible for this colonization resistance? And she was very lucky to find one bacteria, which is called Klebsiella michiganensis, which is from the Klebsiella group, but it's a non-pathogenic bacteria that she was able to isolate and to show that this bacteria restore colonization resistance in antibiotic-induced dysbiosis. And this is part of the data that helped her to demonstrate this. In this case, she colonized mice here in the black line just with E. coli. And then 3 days after, she either introduced another E. coli strain, which is here the blue line, or the Klebsiella michiganensis, and she could see that the Klebsiella michiganensis was able to displace significantly the E. coli that was introduced first. So through multiple experiments, she was able to show that E. coli, that Klebsiella was able to provide colonization resistance, but also displacement of E. coli. So then she studied the process that was involved through this competition between Klebsiella and michiganensis, and she was able. So she used germ-free mice to remove the presence of the other microbes. In this experiment, she was able to she monocolonized the mice with E. coli, and then gave Klebsiella michiganensis that could displace or reduce significantly the loads of E. coli. And then she wanted to study the role of nutrition competition in this process, and what she did was to give the mice a carbon source that only that only E. coli could use. This is here in the yellow line. So by giving a carbon source that only E. coli could use, then she was able to have the mice colonized both with Klebsiella michiganensis and E. coli, so they could now coexist in the same mice, and to show that this competition of Klebsiella towards E. coli could be abolished if by adding an additional nutritional source. And this would not happen if we would give an E. coli mutant that could not use this Galactitol exclusive E. coli carbon source. So nutrition advantage prevents E. coli from being outcompeted by Klebsiella michiganensis, providing support for a nutrition competition as the colonization resistant mechanism. So to summarize a long story short, in this paper published by Rita Oliveira, she was able to identify Klebsiella michiganensis as a protective keystone species to invading pathogens such as Salmonella, multidrug-resistant E. coli, and Vibrio cholera, and that the mechanism involved is mainly through nutrition competition and as a mechanism of colonization resistance. So to finalize, as I told you, we've been focusing on colonization resistance, and we've been showing that current sensing can have a role in this process. We have identified a keystone member of this microbiota and studied the coevolution interactions and how they shape the gut metabolome. This is my laboratory, and most of the data that I show you was from Rita Oliveira and also Miguel Pedro. Thank you all for listening.
Karina, thank you very much. You were so good with time. If there is one question, I think we could allow it, if the organizers agree. No questions?
I can ask one if nobody asks.
Okay, Luis, go ahead.
Just when you have this competition, the titers, I mean, the levels go down very much. They go tenfold down, but the E. coli does not disappear. What do you think is happening there?
Yeah, I think there's still a niche where they're not interacting. I think that's what's happening, and it's a much smaller niche, but then that's probably what's happening here.
Karina, can I ask you just
okay.
When you monocolonize with the E. coli, do you see any signs of inflammation in the gut?
Not with this E. coli. This particular one, it's a very and It's a MG1655, which is very innocuous to the host. But yeah, we've, for example, we've done this with Salmonella, and it also then we see inflammation, right? And because
can you move out the Salmonella with the second microbe?
Yeah. So what happened with Salmonella is that it delays, like for 3 days, it delays the colonization of Salmonella, but eventually Salmonella can enter.
Yeah.
And then, so it really prevents the colonization, but then once Salmonella can reach, it's much slower in reaching its normal levels, but once it does that, then the process goes as if Klebsiella is not there. So it's really at the colonization level that it's acting.